on this worksheet that you picked up just now, there's a fair amount of vocab term that I'm just going to introduce now, and we'll talk a little bit more later. There's this idea of homozygous and heterozygous. We use those terms a fair amount. If you think about the prefixes of homo and hetero, think about everyday terms that we sometimes hear, heterosexual, homosexual, are things that we know in our vocabulary, and think about what those prefixes mean. So when we talk about homo or the prefix hetero, those prefixes, homo means same, the prefix of hetero means different. Okay? Actually, you know what? I see some of you grab that other sheet. We should just, if you didn't, sorry, it's a good idea. Why don't we grab that sheet too? If you haven't grabbed that one, you can grab that. Did everyone grab both sheets? Yeah? Yeah? Okay, well then let's just go through these terms on the board, and then we don't do it twice. That's a better idea. I'm glad you guys came up with that. Okay, so let's start with dominant alleles. So when we talk about alleles are letters, and the letters that we deal with are associated with genetic traits. So we always use the two letters to express a genetic trait. And if it's a dominant allele, it's an allele that covers up or masks another form of that trait. It hides the recessive trait. And we always use the capital letter when we talk about a dominant allele. So when we talk about dominant alleles, we use an uppercase letter, and it dominates over the recessive. It's like King of the Hill. King of the Mountain. You guys ever play that when you were a kid in the school playground? Okay. If you were the king, you dominated over everyone else, and you were shoving them off the snow pile in a very safe manner, of course. And they would try to come up, but you were dominated over them and you pushed them down. It's kind of the same idea. They weren't ever able to get to the top because you were on top. You were more powerful. Same idea with a dominant allele. The recessive allele might be there, but we don't see it, we don't hear it, because it's being dominated by a different allele. Not always the case. We're going to start with the ones that are dominant or recessive. Sometimes there's some in-betweens, but that's what we're going to start with. A recessive allele, on the other hand, is an allele whose trait is only seen when there is no dominant trait present, when no dominant allele is present. And we always use a lowercase letter. Okay? So, it asks for an example, I think. Would be like a lowercase r, a lowercase t, lowercase, pick a letter, would be a recessive allele. What would a dominant allele look like? Pick a dominant, pick a capital letter. That's a dominant allele. The dominant alleles dominate over the recessive alleles. The only time we see a recessive allele is when we don't have a dominant. So we'll get to the kind of the format here of terminology. But now going to the prefixes that I was just referring to, homozygous and heterozygous. The prefix homo, the prefix means same. The prefix of hetero, I don't know why I did all caps. Hetero means different. So when we talk about homozygous, we're talking about alleles that are the same, where heterozygous means our alleles are different. And when we're talking about same and different, what we're talking about is capitalization ones. Okay? Are they both uppercase? Are they the same uppercase? Or are they the same lowercase? This is an example of a homozygous trait, or a homozygous allele combination. This is an example of a homozygous allele combination. The alleles are the same function or capitalization. They're homozygous. Heterozygous means when they're different, so is a big R and a little r. They are different from one another. A big A or a little a are different from one another. They have different alleles. Now, the heterozygous have two different alleles. They have a capital and a lowercase. And capital letters represent dominant and lowercase represent recessive. What are we going to see here? Are we going to see the dominant, or are we going to see the recessive trait? There's someone very dominant in their voice dominates the conversation. There's one who's a little bit more passive with their voice. which one are you gonna hear if people talk at the same time? The dominant, right? Because they're very loud. Okay? If someone's talking like this, it's kind of hard to hear them. But if you're talking like this, you can hear me right now because I'm the only one talking, so you can hear me. But if I'm in the room talking like this, when someone's talking like this voice won't be heard. And that's exactly what happens with these heterozygous. The dominant allele gets masked or gets hidden. I'm sorry, the dominant allele the dominant allele masks. I always struggle with that word, masks. I don't know. You guys struggle with that word? In the morning, it's like, which mask? All right, do we have our masks?
Speaking of masks, I would like to buy some from junior high. You have $10.
Right.
Come on, it's a good cause. It's the business class over there, and Jim's coming over. $5 a piece. Black ones, adjustable ear straps. You want one? I can order you one too.
I have a 20 or I have 8. Or thousands. I have $1,000 bills in here.
Can I have a 20 and I'll try to find change from somebody? Any of you guys have change for a 20? Okay, I'll try.
Or you can bum you from someone.
I'll do that too.
Maybe I show her for it.
Or you don't want it?
I don't know.
Just keep it.
Who cares?
Or change for a 20.
All right. Take 2. All right. I'm not going to cut that out just for the record. Homozygous recessive. So then we talk about these terms called genotypes and phenotypes. When we talk about genotypes are the genetic makeup. It's the genes. It's the letters. These are genotypes. Big R, little r is a genotype. Big A, little a is a genotype. Little r is a genotype. Big R is a genotype. Insert any letters, so we can use any letters we really want. We kind of choose some that are better. Like, it's pretty easy to tell the difference between a big A and a little a, where if I say, is that a capital C or a lowercase c? And you're like, I don't know, you didn't write another one. Okay. Now what? Right, and you're just trying to make a point. You know, so those are kind of hard to distinguish. P's are, unless there's like a line, you know, or they're side by side, then it's pretty easy to kind of distinguish. So sometimes we use them, but we often try to pick ones that are pretty obvious that are uppercase or lowercase letters. So, genotype is the genes, it's the letters, it's what we get when we do a Punnett square. Where when we talk about phenotypes, the phenotypes are the physical appearance, what we physically see, what physically is being observed or expressed. Example could be tall, it could be red flowers. A tall plant could be red flowers, it could be blue eyes. It doesn't have to be maybe something we physically see, but it's a trait being observed. So it could be, I just had it, lactose intolerant would be the trait. It would be the physical characteristic that you have you are lactose intolerant. We don't physically see it, but it's the trait that's being observed or the trait that's being expressed. Okay. So that's what these terms on the top of this are. That's the terms there. That's the terms you wrote on that other sheet of paper. Okay. Let's actually maybe skip this sheet that I have on the board for right now, and maybe we'll come back to it, or maybe we won't. I don't know, because we can kind of accomplish some of the same things. The intent was I was supposed to get to this on Tuesday, but I didn't with you guys. For the rest of you, maybe I did. So let's go through and talk about this sheet. It's called an activator. At the end of today, this sheet should be completed. And under today's in-person agenda, there is an assignment called in-person Punnett Square Practice Activator that I'm going to ask you to take a picture and submit both sides of. We're going to get it done in class. You should get it. Make sure you turn it in. Tomorrow and Monday, your virtual days, there's 2 assignments. One of them will look very similar to this. and it's called Punnett Square Practice Worksheet, and that's under your virtual days. So make sure when you're submitting those, you submit them in the proper spot, and these are 2 different assignments. So if for some reason you get a 0 on one of these, it's probably because, look at the comment, but it's probably because you turned in the wrong one in the wrong spot, or you didn't turn it in. But I'll remind you at the end of class to take a picture of this. So we already kind of filled out the top of this. So genotypes is the letters, phenotypes is what we physically see. So when we go through and we start looking at the bottom of this, there's lots of genotypes listed. These are all genotypes. It's all letter combinations that go along and are being, is that too dark? That's kind of dark. Is it kind of dark in the back? Are you guys okay in the back? Okay, all right. So, so when we talk about these are all genotypes. These are all genotypes. This is a genotype, this is a genotype, this is a genotype. Because it's the genes, it's the letters. But what we're trying to do is we want you to, given a genotype, to tell me the phenotype. I will never ask you to memorize that a purple flower trait is dominant over a white. It's not something I care about, but I do want, given the information that purple flowers are dominant over white, you should be able to determine what we're going to see. So when we talk about a purple flower that is dominant over the white, that means that when I use my letters, P's, we're using P's here, that my P, we use the same letters, big P is going to be the dominant allele, where my lowercase, I'm trying to show that's a lowercase, my lowercase p is going to be the white allele. P's are one of those letters that don't work great, but we can kind of make it. So if we have a big P, where big P is dominant, little p is recessive, if it's big P, are we going to see the dominant or the recessive trait? We're going to see the dominant trait, and because right, there isn't even a dominant trait in there, so to say that you'd see the recessive trait is not very likely. But what we're going to see is we're going to see the dominant trait, and the dominant trait is purple, and technically it's purple flower, but I'm going to be lazy and just write the word purple. What are we physically going to see? We are going to physically see that the flowers are purple. What if it's, I'm going to skip over to little p. If little p stands for white, and we got little p, what are we going to see for flower color? White. What if we have one of each? Now what do we see? We see the purple flower which dominates over the white. Okay? It overpowers it. It covers it up. If hairy knuckles, big H, is dominant, or I'm sorry, if hairy knuckles are dominant, so I'm going to use a big H because it's the dominant trait, and non-hairy knuckles are recessive, therefore that's going to be a lowercase h. Big H is going to be hairy or not hairy? Hairy knuckles. What about little h? Non-hairy. And what about a big H and a little h? We're going to see hairy because hairy dominates over non-hairy. I change the order on you. Bobtails in cats are recessive and normal tails are dominant. So using the letter T's, what are we going to use for bobtail? Big T or little t for a bobtail allele? Yeah, because it's a recessive trait where normal is a big T, right? So we flipped it a little bit. So big T, what are we going to see, normal or bobtail? And if it's little t? Good. Big T, little t, what are we going to see if we have both alleles? We're going to see the dominant allele, which is normal. You try the next two. Round seeds are dominant over wrinkled, and no cleft chin is dominant over cleft chin. Now I apologize because on Tuesday we told you that cleft chin was a dominant trait. Now this is saying it's a recessive. I don't know which one it is. I thought I knew, but then this says something different. It doesn't matter. Based upon this information, fill in this information based upon what was given today. So you try these last two. 10 more seconds. And that's hopefully what you have. A big R, we're gonna see the round dominant allele. Big R, little r, we're gonna see the round dominant trait, I'm sorry. We're gonna see the round dominant trait. And a little r, we are gonna see the recessive trait. Cleft chin we're saying is dominant over no cleft chin. So big C is gonna be no cleft chin. Big C, little c is gonna be no cleft chin. The only time we see the recessive trait is in the absence of a dominant trait. Questions on that before I flip it? Sorry, I was flipping it already. Okay, so now again, kind of using the vocabs of homozygous and heterozygous. Remember, homo means same, hetero means different. If we're talking about homozygous dominant, we're talking bigs. If we're talking about homozygous recessive, we're talking about little. Bless you. Okay? So homo means same. Dominant means we're going to use capital letters. Heterozygous means they're going to be different uppercase and lowercase. And homozygous recessive means they're going to be the same, but they're both going to be little letters. So using those kind of codes, HD, HZ, HR, I'm asking us, we're going to go through and kind of categorize. So if we look at big A, capitalization-wise, are those the same or are they different from one another? Same. And are they bigs or littles? Bigs, which represents dominant or recessive traits. So we would recode that as HD, because that's what this says is HD, homozygous dominant. Big F, little f, same or different?
Different.
So that's going to be HZ. Big A, little a, H. I'm sorry. Big A, little a. Is that homozygous or heterozygous? Let's start there. Are they same or different?
Different.
Therefore, is that homozygous or heterozygous? Heterozygous. And are they the same or? Yeah. Sorry, hetero. So that's the end of that. Heterozygous. Little g, same or different?
Same.
And is that bigs or littles?
Little.
Therefore, is that dominant or recessive?
Recessive.
So that's going to be HR. Why don't you try the next 8? Big G, little g is going to be H. Fill in the last letter. H, Z. Big P is H. D for dominant. Big I is H for heterozygous. Little t is H for recessive. Big T, H, D. Big T, little t is H. Little a, H. And big O, little o. All right, questions on that? Now what we're going to do is I'm sorry, I asked and then I didn't actually wait for a response. Are there any questions? Okay. Now what we're going to use is we're going to kind of use some of that terminology to kind of build Punnett squares as we kind of go. So What is the genotypic and phenotypic ratios? That's a pretty common thing we're gonna ask you to do, is determine whether genotypes and phenotypes are. When we do a cross, so the idea of when we do a cross, we're gonna allow two organisms to reproduce, either actually reproduce or hypothetically reproduce. So what are we gonna get when we get a homozygous tall and a homozygous short? If big T, it represents tall allele, and little t represents the short recessive allele. So homozygous tall, homozygous, homo, the prefix homo means same or different? Same. And if it's tall, is it gonna have the dominant allele or the recessive allele? So it's gonna be a big T, then what's the second letter? Big T, right? It's gotta be homozygous tall. And if it's homozygous short, it's gonna be little t, little t. And I kinda usually do a weird cursive T, I don't know why, but I do. Then this parent is either gonna pass on a big T during meiosis, or they're gonna pass on that big T. So what the idea is, we put a big T on one column, we put a big T on the other column. Now, if you wanted to, you could put them in the rows. It doesn't matter where these get put. Which parent, it doesn't matter if parent one is here or parent one is here, just for the record. And then we do this, we often refer to it as drag and drop. And the idea when we do a Punnett square is we drag and we drop, we drag and we drop, we drag and we drop, and we fill in the possible combinations. So if there's a capital letter in a Punnett square, we put the capital letter first. It's just the way we do it. Is it possible for you to write a sentence without capitalizing the first letter in the sentence? Yes. Do we still get the idea? Yes, but it's not right. Okay, that's kind of the same idea. So big T comes down, little t comes over. We drop the big T, we drag the little t. The big T comes down, the little t comes over, the big T comes down, the little t comes over. Seventh grade, you guys did Punnett squares in seventh grade, right? So this hopefully isn't too foreign, this part. I hope. Now, genotypic and phenotypic ratios, the genotypes are the letter, what are all the different letter combinations that we could get in this Punnett square. Well, I could get a big T, little t, so I'm gonna write that down, or I could get a big T, little t, oh, I already got that, or a big T, little t, well, I already got that, or a big T, oh, yep, I got all the possible genetic combinations. Now, that's just a genotype. We wanna know what the probability is that we're gonna get a big T, little t. And really all this is we look at our 4 boxes, and out of the 4 boxes, I like to write them as fractions. You could write them as a percentage or as a complete ratio if you wanted to, but I often count boxes and I say, hey, it's a 4 out of 4 chance that I'm gonna get a big T, little t. Which really means, right, it's 100%. I'm gonna get a big T, little t. Phenotype, what are they physically gonna look like? Well, this plant has a big T, so that one's gonna be tall. This one has a big T, so it's gonna be tall. This one would have a big T, so it's gonna be tall. This one would have a big T, so it's gonna be tall. So out of the 4 boxes, 4 out of 4 end up being a tall peatland. Or a tall plant, I guess. I said peatland, but it's a tall plant. What if 2 people that are heterozygous for brown eyes were to have a child? So one parent, the parent number 1 is heterozygous, parent number 2 is heterozygous. If we use big B for brown and little b for blue eyes, oops, sorry. What's the genotype of parent number 1 gonna be? Heterozygous, hetero means? Same or different? Different, so it's gonna be what? Big B, little b, and the second parent is heterozygous, so therefore it's gonna be big B, little b. Okay, so parent number 1, big B or little b, parent 2, big B or little b. And then we pull these down and across. Big B, big B. big B, little b. Big B, little b, little b, little b. Again, if there's a capital letter, we write that first. So we go through and we say, what are all the different letter combinations? I see a big B, big B. I see a big B, little b. And I see a little b, little b. Out of these 4 boxes, how many are big B, big B? 1. Out of these 4 boxes, how many are big B, little b's? 2. Now you could either write this as 2/4 or 1/2. If you want to reduce the fraction, you can. If you don't want to, you don't have to. And how many are little b, little b? We get 1 out of 4. Phenotypically, what's this one gonna be, brown or blue? What about this one?
Brown.
What about this one?
Brown.
What about this one?
Blue.
So how many are gonna be brown? 3 out of the 4 are gonna have brown eyes. 1 out of the 4 are gonna have blue eyes. Phenotype, physically look. How do they physically look when we talk about phenotypes? Why don't you fill in, based upon the information, using big A for purple, little a for white, what would be the parents' genotypes if you're gonna cross a homozygous white and a heterozygous purple? Let's just do the parent genotypes first. I'll give you 10 seconds to do the parents' genotypes based upon homozygous white, heterozygous purple. Don't do the Punnett square yet. All right, so if parent number 1 is homozygous white, what's their genotype gonna be? Little a, little a. Why? Because they have the white allele, that's recessive, and it says homozygous. Now, what if I were just to say a white plant? What's the genotype of a white plant? Right? If I were just to tell you it's white, the only possible genotype that can be white is little a, little a. Okay, just be aware of that. Heterozygous purple, I don't need the purple, I could have just said heterozygous, but regardless, heterozygous means big A, little a. Now try your Punnett square. Just for the record, I'm gonna put my big A on top, but you can put it wherever you want, it'll just get rotated a little bit. So you try to fill out your Punnett square. So my Punnett square looks something like that. Because I put my big A here, my big A's are in this first column. Now, if you were to put your big A here, just realize your 2 big A's would be in this row, but outcomes end up being the same. Why don't you try to fill out your genotypic ratios? What are the possible, all of the possible genotypes that you could list? And then how many out of the 4 are gonna be like that? So our genotypic ratios, 2 out of 4 or 1/2 of them are gonna be big A, little a, and 1/2 or 2/4 are gonna be little a, little a. Purple versus white. What color is this one?
Purple,
What about this?
purple,
What about this?
white, white,
Okay, so of these 4, how many are gonna be purple? How do they physically look? We're gonna have half, or 2 out of the 4 are gonna be purple. And of the 4, half, or 2 out of 4 are gonna be white. How do we feel about those? Thumbs up? Yeah. Thumbs kinda. Thumbs down? I don't know. Okay, so some of you didn't vote, which is a little concerning. So, your homework for today was to go through section 1 notes. We are going to skip section 2 because we kind of just did section 2, so there is going to be no section 2 notes. One of your homeworks for your 2 virtual days coming up is going to be section 3 notes. I'm going to talk really brief about section 1, talk briefly about section 2, and then we'll kind of see where we're at. So, section 1 talked about the blending hypothesis. Unless you listened to the recording, you probably didn't hear what I'm about to say. But the idea of this blending hypothesis is what was thought to have been true prior to Gregor Mendel going through and doing his work with pea plants. This idea that was observed was sometimes a red flower and a white flower, if they were to pollinate, would produce a pink flower. That happens. It does occur in certain plants. So the idea originally thought, Mendel disproved, the original idea was called the blending hypothesis. And the blending hypothesis basically say we take the 2 traits and we just kind of mix them all together. So why do you look the way you look? Because your mom and your dad's genes are all mixed together, and that's why it is. Okay, purple and white and red make pink, kind of supports that. And here's another thing that kind of supports it. Again, Mendel disproved it. But if we were to take a blue pitcher of water and a yellow pitcher of water, and we were to mix those 2 together, what would we see? Obviously, it's right here that we clearly can see. What color of water are we going to get as a result? Blue and yellow make green, right? So now, if we were to take that green water, and we were to take 2 pitchers of green water and pour it together, what would we get? Green. And from there on out, forever, we would always get green water, right? Does it ever happen where we take blue and yellow, we mix them and we get green, and then we take those greens and we mix them and we get a yellow or a blue back? No, right? They're blended, they're permanently mixed. However, with these flowers, if we take a white and a yellow, red and a white, certain species of flowers will produce pink flowers. If we take a pink flower and a pink flower, we get some pink flowers, which would support the blending hypothesis. However, in some species of flowers, the whites sometimes show back up and the reds show back up. That'd be like pouring green and green and getting blue. It doesn't happen. If the blending hypothesis is correct, that should never occur. However, it does happen. If I mix a pink and a pink, I do get the white to come back. I get the red to come back. Mendel disproved the blending hypothesis. Gregor Mendel, Austrian monk, did a lot of experimentation and data collection with pea pods. Very conveniently, he picked pea pods, or pea plants, I should say, because they had 7 traits that are what we refer to as completely dominant. The pea plant was either going to be tall or it's going to be short. The pea pod was going to be green or it was going to be yellow. The flower was going to be white or it was going to be purple. The pea was going to be round or it was going to be wrinkled. There's nothing in between. That's not true with all traits. A lot of our traits are controlled by many different combinations, but pea plants aren't. So again, kind of a nice, convenient thing that he picked that. And what he found was a couple things. He found that certain traits showed up more prevalently or more commonly than others, and he deemed those dominant traits versus recessive traits. And he also found that there was no connection between the color of the peas and the color of the flowers, or the flower colors versus the color of the pea pods. Those were separate genes that were controlled by separate things. Okay, and that's what Mendel kind of came up with. And he went to great lengths to kind of work through this. This is kind of like the situation, I think the problem we did, if we were to take a purple and a white flower that he called true breeding, and he went to great lengths to make sure he had true breeding flowers, and when he crossed a purple and a white flower, he got 100% of his plants were purple. But if he took, if we kind of think of ancestry, this is like your grandparents, this is your parents, this is like you and your brothers and sisters. If the parents were purple, grandparents were purple and white, all of the parents' generation were purple, but some of your brothers and sisters are purple, and one of you, statistically, was white colored flower. So that white flower, that recessive trait that was hidden or masked in the parent generation or the F1 generation, showed back up. Okay, so we kind of had this idea of dominance. Again, great lengths. He cut off the pollen producing things. He covered them with like bags so they wouldn't pollinate themselves. But the idea goes back to kind of meiosis. When we were going through meiosis and we drew those chromosomes, we drew like a red chromosome here and a green chromosome here, and I talked about how these two align is random. You know, is it green red or is it red green? And what happens is that's where these traits come from. In this particular flower, the male part of the flower is referred to as the pollen. Had a big B or a little b, but which one got passed on in that pollen, we don't know. There's no way to predict it. It was either going to be this one or it was going to be this one. The pistil part of the flower was either going to be a big B or a little b. We don't know which one. It's a random, how these align. We can make predictions, but we can't guarantee results. And these are called alleles. So a big B is the dominant allele, little b is recessive allele, and we can go through and we can do Punnett squares with these. Now, all of these Punnett squares, really all they do is they predict a possible outcome. It's a probability. If I were to flip a coin, what's the probability that the coin I flip is going to be a heads? 50%, right? And what if you flip a coin? What's the probability that the coin that you flip is not the same coin, it's not my coin, it's your own coin, that you flip your coin, that you're going to flip a heads? 50%. What if I flip heads, and now I ask you to flip a heads? Does the fact that I just flipped heads have any effect on what you flip?
No.
They're independent events, right? Now, if I get heads, so 1 out of 2 chance, or 50% chance that I'm going to get heads. It's a 50% chance that you're going to get heads. What's the probability that you and I both get heads? 25%, right? And every time we flip a coin is a new random event. So if we were to flip a coin 3 times, the probability of getting heads each time is 1/2. So 1/2 times 1/2 times 1/2 would be 1/8. Okay, is a 1 out of 8 chance that 3 flips in a row would be heads, heads, heads. Now, if I flip heads 3 times in a row, that 4th flip, what's the probability that one's going to be heads?
1/16.
Just that one flip. What's the probability that, what's that? It's going to be 50%. None of the 3 prior flips have any effect on the next one. Right? They're all, past outcomes do not impact future ones. Past outcomes do not impact future ones, if it is a random event. I used to use slot machines as an example of this because I thought this is how they work, but I found out it's not. I used to think that every time you pull the lever or push the button on a slot machine, it's a random event, but it's not. They have like scheduled payouts, so X many coins go in, X many coins have to come out, so it's not a randomized event. Okay, so there is a little bit to be said about like the machine is due. I'm not saying you should go play the slot machines because you're going to win, but the idea is it's not a completely randomized event, but flipping a coin is. Which allele is going to get passed on is a random event. So if you have brown eyes, or your dad has brown eyes and your mom has blue eyes, and dad is homozygous dominant, right? Homo, same, dominant. Mom is homozygous recessive, homo, same, recessive, lowercase. Which allele is mom going to pass on? We don't know. It's a flip of a coin, random event. I don't know if she's going to pass on this one or this one. Which one's dad going to pass on? I don't know. This one or this one. It's a new flip of the coin. So I don't know if I'm going to be this one, this one, this one, or this one. Now in this case, what's true about the outcome of all 4 of these? They're all gonna be big B, little b, they're all gonna have brownness. However, that's not always the case, and this would be an example. Now this is talking about flowers, but let's just pretend, this one is what I meant. These colors represent something else, these letters, but let's just pretend like this is a red hair allele, little r, it's recessive, where brown hair is a big R allele, making stuff up, okay? So big R, little r, brown, red, brown, red. What was, if this is dad, what was colored hair did dad have? And what color hair did mom have? Brown. If your parents were to have 4 children, statistically, how many of those children should have brown eyes?
4 is too many, don't get more than 2. I'm kidding, I'm 1 of 5, guys. Just gonna show it off, my new math.
Oh, here.
Where's the junior high business classes?
Wow, everyone, isn't that a really nice face
Is this
mask?
still the same class? You've
Yeah.
been in this class for forever.
Yeah.
I feel like you're telling me. It's kind of big, it would even fit your big face, I think, if you wanted.
Is this some random stranger who borrows money from me? I don't know who it is.
You can get back to it. I'm sure it's super important whenever you use it.
Thank you, Mrs. Davies. So if you were gonna have 4 children, the probability, I'm sorry, if your parents had 4 children, statistically, how many of those children would have brown hair? 3. Statistically, 3 out of the 4 would have brown hair. Now, is it possible that the first child to have red hair? Yeah. What was the probability that that child was gonna have red hair?
I think it's 1 out of 4.
1 out of 4, right? I'm sorry, I don't know what color I said. Red hair, I meant red hair. Red hair, I pointed at this one, but I think I might have said brown. So let's start over. What's the probability that the first child born was gonna have red hair? 25%, 1 out of 4. If they have a second child, does the first child's hair color have any impact on the brother or sister, right? So what's the probability that that second child was gonna have red hair? 25%. It's a new random event. Okay, what about the third child? And the fourth child? Is it possible that 4 out of 4 children could have red hair? Yeah, it's possible. Now, it ends up being like 1 out of 256 chances that all 4 would have red hair because it's 1/4 times 1/4 times 1/4 times 1/4, but it's possible. It is possible. Okay, it's not super probable would be the term we use, but it is possible. Just like I could flip a coin 100 times and get heads 100 times. It's possible, not very probable, right? But that's the idea with these Punnett squares, is they are a prediction of an outcome, not guaranteeing an outcome. Each time we do a Punnett square, each time a fertilization occurs, it is that same Punnett square over again, and the past outcomes does not impact the future. Vocab terms we talked about the idea of theory of segregation is just that these things kind of pull apart and they separate during meiosis. Mom's either gonna, dad in this case, because that's the pollen, so dad is either gonna pass on the big B or the little b. Mom's gonna pass on the big B or the little b, and we don't know which one it is. It's the flip of the coin every time. Okay? It's gonna be big B. Now, that's if it's a heterozygous. If it's homozygous, you got a 2-headed coin then, right? Or you got a 2-tailed coin. You flip a coin that's got 2 heads on it. What's the probability that you're gonna flip heads? 100%, right? If you had a big B, what are you gonna pass on to your kids? Because that's the only thing you have. Just like a 2-headed coin only has a heads. Okay, same thing would be true with what a little b will be. Okay, how do we feel about those terms? A lot of vocab here. We okay with that terminology? Okay, picture. I told you should take a picture at the end of the class of that worksheet that we went through that is referred to as Punnett Square Practice Activator. Okay, it is listed in your, under today's in-person agenda, Punnett Square Activator. Okay, now make sure you put that under the one that says activator. There's 2 sides of it, so make sure you take a picture of both, or I'd appreciate it if you take a picture of both. Then, last thing I want you to do is, on your way out, or right now, there's a stack here, there's also a stack there. There's 2 homework assignments for your virtual days. One is called Punnett Square Practice Worksheet, which, as I hold up, looks very similar to the thing that you just filled out as the activator. There are differences, yes, there are differences. I would encourage you to do this from memory and then check based upon your activator, and then once you get to the Punnett Squares, there are different Punnett Squares. Make sure you submit that in the proper spot that says Punnett Square Practice Worksheet. It's on your virtual day. There's also section 3 guided notes. Those are your 2 homework assignments for Friday, Monday. They're either there, they're there. Wipe your desk. Have a beautiful and wonderful day.